Welcome students. Today in this video we are going to discuss Megasporogenesis. Previously we had discussed microsporogenesis. Megasporogenesis is the phenomena or the process of development of the female gamete, and a female gamete lies inside the female gametophyte, which is known as the embryo sac. So here in the diagram 2.7 of your NCERT book, you can see the A, B, C and D. This D is showing the primary structure of an ovule, and ovule we know is located inside the ovary. This ovule has a mass of undifferentiated cell which is known as nucellus, and inside the nucellus, in the next following diagram 2.8, you can see that a single cell is specialized towards the micropilar end, and this cell is known as megaspore mother cell. This megaspore mother cell undergoes a meiotic division and forms a group of 4 cells which is known as megaspore tetrad. You can see here in the diagram A that is depicting the formation of megaspore dyad, and thereafter it is forming the megaspore tetrad. So the 4 haploid cells of megaspore tetrad is formed, and thereafter 3 of them get degenerated, and the remaining 1 cell remains viable and it develops, it grows into size. Because the cell has been formed by the meiotic division, so all the cells of tetrad is haploid, but the 3 is degenerating, so the remaining haploid cell is forming the embryo sac. It develops, grows in size, and the nuclear division by mitosis takes place inside, forming a group of 8 cells. This is called 8 nucleate stage of the embryo sac. These 8 cells get shifted to opposite pole, and out of group of 4 nuclei at each pole, a single nuclei from both the poles shift to the center. Here in the diagram 2.8 in B, you see here that 8 nucleate stage is there, and these nuclei have been shifted to opposite poles, 4 in group, 4 in number at each pole. Out of these, 1 from each pole is coming or shifting to the center, forming the polar nuclei, and the remaining in the diagram C of 2.8, you can see here the group of 3 cells at each end, that is micropilar end and chalazial end. If there is any confusion for you people, you can see once again the diagram 2.7D, which is showing the primary structure of an ovule. Here you can see that the embryo sac is enclosed by a single or double layer of integument, and integument is later on forming the